Hello, everyone, and welcome to today's webinar, Developing Genotyping Platforms to Enable Discovery, Quality Control, and Genomic Breeding at CoverCress Incorporated. I am Cassie Soltman of LabRoots, and I'll be your moderator for today's event. Today's educational web seminar is presented by LabRoots and brought to you by LGC Biosearch Technologies. To learn more, visit www.biosearchtech.com. We encourage you to participate today by submitting any questions you may have during the presentation. To do so, simply type them into the Ask a Question box to the left and click Submit. We'll answer as many questions as we have time for at the end of the presentation. You may also use the Ask a Question box to let us know if you're having any trouble seeing or hearing the presentation. There will also be a brief poll concluding the presentation. When you are prompted to do so, please take a moment to interact and participate. I'd like to now welcome our speaker for today's webinar, Dr. Ratan Chopra, Vice President of Research at CoverCress Incorporated. For a complete biography on our speaker, please visit the Biography tab on the top of your screen. Dr. Chopra, you may now begin your presentation.
Thank you for the opportunity to present about CoverCress story today. I'm Ratan Chopra. I co-lead product development efforts focused on crop design that enables agronomic features that will benefit farmers and the meal and the oil qualities that will be useful in downstream applications. Today, I'll be discussing about the genotyping platforms that we've developed in collaboration with LGC. And before I jump into that topic, I would like to highlight some of the advancements that we have done with this crop and why we are working on this crop to begin with. So the world is challenged with one of the challenge today is greenhouse gases. And we know that this is coming mostly from burning fossil fuels for energy purposes. There are a lot of strategies in place today by using alternative energy, but there's no perfect solution. And we believe that shifting towards a low carbon feedstock would be one of the most desirable solutions in the near future. While today you could use soybean oil to meet this renewable diesel demand and create renewable diesel, it would create challenges for the existing biodiesel and food customers that use soybean oil. In fact, if you start matching the needs today, you would need 17 billion pounds of soybean oil in addition to what's being produced. So this is a challenge, and the only way we can overcome this is by growing new feedstocks on the same existing land during the off season and create an opportunity for farmers to have extra income at the same time, produce an energy, a source or a feedstock that will be useful in the renewable diesel industry. And the other challenge that's faced in the US primarily is the challenge with the water quality issue and the soil quality issue. You can see on the graph on the left that US agricultural system, where there is a primary focus on corn, soybean rotation, and as you move south, some of the other cropping systems, along that Mississippi River, you see a challenge of nutrient leaching. And this is not a new problem, it's a century-old problem. And what this has done is caused a lot of well water to be in an undrinkable state today. And researchers have performed studies that show implementing cover crops in the off season, as you can see in the picture on the bottom right, will help us reduce that nutrient leaching issue that we see. But the problem is only 3% of the corn and soybean rotation today use some sort of cover crops during this fallow period. So we really need a system and a crop that can be easily adopted and incorporated in these traditional cropping systems and provide the right incentives to the farmers so that we can not only overcome these challenges, but improve the soil quality, reduce the soil erosion, potentially long term improve carbon sequestration and reduce the carbon footprint. To do this effectively, over the last couple of decades, there's been a lot of efforts from federal agencies like USDA and Department of Energy funding towards these academic institutions to think about alternative energy sources and feedstocks. And what that has led to is forming of these groups or subgroups that are primarily focused on identifying crops that would be well adapted to the specific regions of the country and could serve as the cover crop during the off season, which would feed into the feedstock industry, at the same time provide all the benefits that we are looking from an ecosystem services standpoint. Just to highlight a couple of them, I Prefer is one of the one which covers a lot of I states and Minnesota and Wisconsin, primarily focused on domesticated pennycress or covercress, whereas Spark, southeast of US, focused on Brassica carinata. And there's Camelina sativa and other winter oilseeds like winter canola. And there have been some efforts towards identifying cellulosic feedstocks that could help address this demand for renewable diesel and overcome some of the challenges that we see with the gas emission or with the nutrient runoff. Without further ado, I want to kind of talk about what is CoverCress and how we are using this crop as an opportunity to solve a couple of problems that I described earlier. CoverCress is a new winter oilseed crop that is primarily been focused towards non-feed and renewable diesel industry. And it has been over a decade of research between academic partners and CoverCress to really use every tool that was available at hand, whether it's a traditional breeding efforts or mutagenesis or gene editing, to create this field pennycress to a cash cover crop known as CoverCress as a new winter oilseed crop for the upper Midwest that could be incorporated in these cropping systems. And what CoverCress does is really provides an opportunity to address the challenges that I discussed early on and provides an economic opportunity for farmers. For example, it provides the benefit of cover crops by improving the soil health, retaining the moisture, reducing the nutrient runoff, and sequestering carbon that is limited during the off season because there is not a lot of photosynthetic activity occurring. At the same time, you think about two cropping systems of the United States, corn and soybean in the Midwest, and adding another crop really makes it a three cash crop system, which provides economic benefits for the farmers. And what we've focused is trying to avoid using any new land and equipment to generate this crop on the existing land while reducing the carbon intensity score. To support all of this, there is another important thing that we focus on is trying to mitigate risk for farmer adoption by using a closed loop business model where we provide the grain, we buy the grain back and move it down the supply chain. And this is possible today for us while there's been a lot of research and we've identified what the potential value for this crop is. It would not be possible without a strategic partnership that we have or owners we have in place today that help us guide in the right direction with their experience in different parts of this supply chain, Bayer on the upstream, focusing towards product development and the growers, and in the midstream, Bunge really focusing on crushing aspects of this grain, and downstream, oil processors using this oil to be used as a renewable diesel. So this has been very helpful partnership that helps us guide in the right direction to bring this new crop to the market in a very short time. Finally, how do we see this crop being scaled or adopted in the Midwest agriculture? As you can see, our primary goal is to meet the demand of renewable diesel market, and there is a lot of land that's available during the off season highlighted here in the map. um, our primary target today is just that green highlighted region, and primarily because we want to focus on getting the adoption right and getting the right engagement with the farmers. Even the way we think about scaling of our acreage, we think we can address about 3+ million acres by 2030, 2035, if we can focus primarily on that potential market, which is targeted towards producing grain that would move towards renewable feedstock in the near future. So with that background, I'm going to switch a little bit here and talk about what really is field pennycress and what has happened over this decade to kind of bring this to a market as a crop. And then I'll highlight onto some of the traits and then get into the genotyping platforms. So field pennycress is a brassica species. It's also known as Thalaspi albans. It is believed that its origin is in Eurasia. It's naturalized to a lot of the cooler climates of the world, northern hemisphere or southern hemisphere. You will find this in disturbed soil near the agricultural land. It's not invasive species. And typically you find two growth habits, a winter and a spring growth habit. A winter growth habit generally establishes in the fall and then flowers sometime in March or April, depending on the geography. Whereas spring annuals tend to establish sometime in March and then flower in April and then transition quickly. While our focus today is winter annual form because we want to get the winter cover crop benefits and provide a feedstock that is going to be of low CI score. Pennycress can be planted, or domesticated pennycress can be planted, and average yields of 1,000 to 2,000 pounds per acre can be achieved depending on the geography. And the seeds contain 30 to 36% oil and 24 to 28% protein depending on the varieties. And just the yield and the oil makes it more attractable for renewable diesel industry. As for any crop, it is important when you think about crop improvement to have genomic resources. And what's been done over the last 10 years is building these genomic resources. That's happened at institutions like University of Minnesota, Illinois State, and CoverCress, where they've been able to identify what it is. It's a diploid species, it's a self-pollinating species. The genome size ranges from 500 to 540 MB. It's got 7 chromosomes. There's been 2 versions of the genomes over these years. The first draft genome had a lot of scaffolds, smaller scaffolds, but the more recent assemblies have shown chromosome level assemblies. The gene space have been established. Like there's a good indication of methylation patterns, the small RNA patterns, and tissue-specific expressions. And very recently, there's been an effort by iPrep group really focused on sequencing accessions that have been globally collected and shed a light on what's happening in the natural variation among the genotypes or lines that have been collected for this species. So a lot of genomic resources have been built to really shed a light on genetic architecture and composition. But that's one piece of crop improvement. The other piece of crop improvement is breeding, as there was a realization that this crop could be domesticated and used to meet the fuel needs. There were a lot of efforts made starting in 2009 all the way to 2014. Different breeding programs got started. There was a lot of germplasm collection that happened at these institutions, whether at CoverCress, Western Illinois, USDA Peoria, University of Minnesota. And the germplasm collection continues to happen by Dr. Sedbrook. But the idea there was to take these collections, develop varieties that could be agronomically desirable for farmers to grow, get the production that would be desirable for making that extra trip to the field. So in that process, there's been a few public varieties that have been released in the past. And CoverCress has identified germplasm, commercial germplasm varieties that are going to field can yield up to 1350 pounds per acre. And we continue to think about building this yield trait into our future products with the anticipated goal to reach 2,000 pounds per acre yield. While genomic resources have helped us identify the gene space and the variation among the germplasm, and the breeding has really allowed us to make the gains or enhance the yield or the performance of this crop. One of the important things that happened during this 10 years was identifying the nature of this species compared to Arabidopsis. And this was primarily seen, observed under Dr. Mark's supervision at University of Minnesota, and then joined hand with Dr. Sedbrook at Illinois State, where they found that pennycress is very similar to Arabidopsis. In fact, the gene space among these two species was so highly conserved, as you can see at the bottom of the slide, that the gene space or the orientation of the genes was very conserved. And this understanding really allowed quickly for scientists working on pennycress to leverage Arabidopsis research and think about translational research and identify traits that would be valuable in domesticating this crop. For example, just a figure on your right, focused on the seed color or the anthocyanin biosynthesis. Using this knowledge, we were quickly able to identify mutations causing variations in the seed color and point to the genes that were already characterized in Arabidopsis. Now, this is not going to be true for every gene or every trait that you think, but it has been observed again and again that you can leverage a lot of information from Arabidopsis and make significant gains to bring this new crop to the market. And what's happening because of this translational aspect of the species and the potential to be a new crop in decades, there's been a lot of growing interest in research labs across the country where we have started seeing researchers really focusing on using this species to understand the biological questions that they were not able to answer using Arabidopsis, or provide new value to this crop as it comes to the market. Finally, you think about resources, gene space, breeding efforts. What's important is you need the right modern tools that would further accelerate deployment of traits into the species that would be desirable for downstream applications, whether it's agronomic establishment or for downstream application of the meal or the fuel or the oil quality aspects. In 2016, Dr. Sedbrook at Illinois State University was able to establish protocols for transformation and showed that editing can be achieved in pennycress. And this transformation is very similar to Arabidopsis, a floral transformation. And over the years, CoverCress has optimized the system, have identified some fluorescent markers to really further increase efficiencies in transformation and also selection of transformants that would go on to be edited in the program. And today, with the transformation tool and the editing capacity, we are able to convert a line in less than 9 months with multiple traits of interest or genetic edits in a single line. And use of fluorescent markers and floral dip method have really been crucial to make the changes that we have been able to make in this new crop. So with that information on the tools and the species and what has been done over these last 10 years, we've reached this point of domesticated pennycress, and that's what we call CoverCress. So domesticated pennycress has features that makes it non-weed species. It reduces the need for post-harvest management that would exist with the wild pennycress. And what I'm going to do is I'm going to spend some time and highlight some of those traits and kind of point to some of the features that we have changed that allows us to have reduced weediness characteristics in CoverCress grain. So the first one is low fiber trait. So the low fiber trait is a modification to the seed color biosynthesis pathway, as I showed earlier, anthocyanin pathway. There's been a lot of genes that have been characterized in pennycress now, where upon editing or through mutagenesis, you can get this yellow seed color. And what this does, it quickly reduces the fiber, improves the fiber quality, which is desirable for feeding animals and makes more energy available in the diet. When this project came into play and we saw this change in color, it benefits the fiber quality or the fiber content of the meal. As we started looking carefully into other aspects of this seed or the meal, we found, in fact, a change from black seed to yellow seed resulted in higher protein content, a higher oil content, and an ability to germinate more uniformly compared to wild pennycress. For example, wild pennycress, you would see about 50 to 60% germination without GA treatment. But with CoverCress grain, with this yellow seeded trait, you can get 95% germination right after planting. And what this trait has allowed us to really overcome some of the challenges for feeding, but also help us overcome the weediness issues that are observed with the wild pennycress. And just to highlight a few, there's a lot of features, a lot of studies that we've done on this material to see what we've achieved with this trait. First thing, increased germination and reduced dormancy. We've seen again and again, you have a black seed untreated with GA, it takes a longer time to establish and also fewer seeds germinate. Whereas yellow seed, you consistently see over 90% of the seeds germinate. And it's been reported in a couple of papers now as well. And we've further collaborated with Western Illinois to understand whether the seed longevity decreases with this yellow seed. In fact, what we've seen with the seed burial test, a graph on the top right, shows that black seed on the left, despite being buried under the soil for several days or months, can be vigorous and can establish, whereas the yellow seed quickly rots and loses the viability. Finally, we've also looked at the viability under stress conditions. And to do that, we did accelerated aging studies. And what we've seen is accelerated aging would destroy the seed quality of the yellow seed, whereas the black seed would persist despite those harsh conditions. So, all in all, this trait helps the farmers with reducing that weediness characteristics and reduces the need for management post harvest. And with any remaining seeds in the field, because of viability challenges under stress conditions, and it would be subject to summer heat, most of the seeds would get destroyed in the same season and would not carry forward in the next season. This brings me to the second trait of weeds, right? Shattering. Shattering is a common phenomenon in weedy species, and that's primarily to maintain and persist its population in the wild. And as we think about this crop in farmers' field, we want this trait to be there not only to reduce the persistence or dispersal of the seeds, but also increase the yields from the production. While there is a lot of literature that suggests that you could use inheritance gene to create the variation and achieve reduced seed pod shatter, and it's been shown in Arabidopsis, pennycress, and canola, it is always challenging to achieve this using gene editing. While you could use backcrossing strategy to the alleles that have been identified in pennycress, it is not desirable when you have modern tools like editing to quickly create those changes and have reduced shatter phenotype. Over the last couple of years, we've actually identified a gene that can be edited and we can achieve reduced shatter trait. You can see on the bottom right graph where we've found a mutation that will reduce the shattering significantly in our variety. And we continue to evaluate this mutation. We are deploying this in our products, and it'll be fixed in most of our future products, which will be desired for even harvesting and extending the maturity or harvest windows for the farmers. As you're asking farmers to change their practices while they're harvesting, they need to be planting their typical summer annuals. So having this trait will provide some flexibility to farmers as well. So with these two traits that I talked about, reduced fiber or reduced pod shatter, we believe we've significantly reduced the challenges of weediness that exist in the wild pennycress. Couple of other traits that we've continued to think about changing or have changed in oil and meal. First, I'm going to talk about oil. As we think about this product movement in the supply chain, there's a lot of utility of this crop in addition to the renewable diesel industry. The oil can be useful for other applications, but pennycress in its wild form has a very high levels of uric acid in its oil, 35 to 40%. And this particular fatty acid is not suitable for feed or human consumption. And one of our focus for the business has also been towards moving this grain towards poultry industry, which means uric acid at a high inclusion levels could be detrimental for feed. To facilitate that application, we've got rid of this trait, and this was identified at University of Minnesota, Illinois State through mutagenesis and through editing, that you can edit a single gene and achieve this low uric acid trait. As we've identified this solution, we've started incorporating this trait into CoverCress product to enable the feeding of the whole grain to the chickens. And the other compound that we've been working on is reducing the synegrin. Synegrin is a type of glucosinolate that is present in pennycress. Glucosinolates are secondary metabolites present in most of the brassica species. They can be detrimental for feed applications. Specifically, the one in pennycress has a lot of implications on the feed industry. And for us to have good utility of the meal, we want to get rid of this compound. And what we've done over these 5 years is really identified several gene targets that could help us reduce the synegrin in the seed. And we found some other solution that could further decrease that synegrin in the meal. We've found several combinations. The graph on the right shows some of the lines that we have in the pipeline today have that glucosinolate levels below 30 micromole, which is what is desired by FDA standards. And as we are identifying these solutions, our future products will have low fiber, reduced shatter, low uric acid, and low synegrin in the CoverCress product, which will make it desirable for fuel, feed, and also agronomically suitable for farmers and reduce management challenges. So those are the traits that we will have in CoverCress products in the near future. Well, we've made significant progress in identifying the tools that are necessary for bringing this new crop, and we know the application of this crop and have identified traits that will be crucial for initial commercial activity of this crop. What we've been doing is thinking about what does the future look like for the crop improvement or CoverCress improvement. And one of the needs is having the right genotyping platforms. And this is the case in any crop development industry. You need a genotyping platform that helps you meet the needs at various levels. In our case, we really think that genotyping platforms are critical to the success of current and the future products. There's various ways to look at it. Like you can do whole genome sequencing that can provide you genome-wide variation. It can provide you the gene space. You can look at de novo assemblies and really understand all the patterns that might be in the exonic region or non-exonic region. But then you can scale it down further by using targeted resequencing like GBS, where you still are able to observe variation among the germplasm or genotypes, but really focused on a handful of markers represented throughout the genomes. And then another step down would be really looking at a platform like FlexSeq, where you're really looking at markers that can be repeatable, consistent across genotypes and year after year. And today we sit in a place where we have all these platforms established and help us provide solutions for gene discovery, understanding genetic diversity, perform quality control of our germplasm, of our products, perform mapping, and eventually enable genomic breeding in the program. And this is important for any crop improvement. Like I've already mentioned the utility and the application, but one of the things that we think about we use editing as one of our platforms is being able to find the right protospacer sequences that can help you edit multiple products. And these genotyping platforms like whole genome sequencing allow you to do that by designing pan guide RNAs or pan guide protospacer that can be used in multiple products as they come online. You need allele specific marker when you start thinking about breeding in specific mutations using backcrossing strategy, or you want to select certain traits in the F2 generations. And that we've been able to successfully execute using Cas-based technologies in the program or PACE technologies. And finally, we think about the fingerprinting. I already mentioned about quality control, but we want to be able to perform our pre-breeding work or discovery work that will help us create or retain genetic blocks that will be valuable for performance in our future CoverCress product. So that brings me to what we've done in collaboration with LGC over the last year and a half. In the past, we focused on developing GBS as a platform for genotyping our germplasm and help our understanding with the breeding diversity. And when we did that, we noticed that the GBS platform that we used in early on limited our ability to really capture variation because of methylation patterns that we see in pennycress. And it really resulted into low marker density or SNP islands or SNP deserts in most of the chromosome. And this was not encouraging because we wanted to have a cost-effective approach, at the same time capture variation across the chromosome to inform breeding and diversity analysis. During that time, we collaborated with LGC, and the scientists at LGC helped us consider some other enzymes and library prep methods that could facilitate or help us improve our observations on the frequency of SNPs throughout the chromosomes and find good marker density. To do that, we tested about 200 genotypes and sequenced them using the enzymes that they nominated, and then we used certain library prep methods nominated by them to see if we can capture variation. In fact, that allowed us to capture anywhere between 50 to 80,000 variation among the germplasm that was subject in this pilot scale. That was really encouraging, and we were able to overcome the hurdle of GBS that we had seen in the past and capture the variation that we needed to capture in our germplasm or the program. But that's a lot of markers to be capturing in a breeding program or in the QC process of our product development. So we decided to continue our collaboration, and we worked with Rapid Genomics, who's now part of LGC Genomics, to really identify targets that would be enough for us to capture the genetic variation or perform QC. And to do that, we identified about 5,000 regions of interest using that GBS data that was generated and sequence it using Rapid Genomics effort using FlexSeq approach. And what that allowed us to really validate the findings of GBS, but also repeat the markers that we saw in GBS consistently across different germplasm that we used in the validation set. This was encouraging, and then we started thinking about how can we use this in the application of CoverCress breeding program and CoverCress product development. So the first application was really to use this FlexSeq data to see if we can generate a genetic map that would inform breeding and straight tracking decision. As I mentioned earlier, pennycress genome has been characterized, but what has been interesting is the gene space distribution and the methylation patterns are quite unique in pennycress, which always made it challenging for us to understand the recombination in pennycress, and we were unable to generate a linkage map. But using this panel or FlexSeq data, we were able to genotype over around 136 F2 lines derived from a cross between 2032 and MN106. and find about 3,500 markers that were distributed uniformly across the chromosomes, 7 chromosomes, that segregated in Mendelian fashion. We took those markers and really identified the mappable markers, which resulted in this genetic map with the genetic distance of 865 centimorgan. This is one of the first maps today for pennycress that's being generated at CoverCress. It really is valuable to inform the breeding and our crossing strategy as we think about bringing in edits, crossing them with the breeding varieties. This map can be used for mapping the traits that we saw segregate in the F2 population, and the work is ongoing. We will continue to build on this resource, but what's exciting is being able to have a recombination map that allows you to locate and understand the frequencies would inform the crossing decisions that are very critical when you start thinking about stacking multiple traits, multiple genes into one product without creating linkage blocks that might be detrimental for performance. That was one of the applications. The second application that we've been quickly able to apply using this panel is to understand the genetic diversity among the germplasm and the breeding population. As I mentioned, we targeted 5,000 regions. And when we targeted those 5,000 regions, we were actually able to identify about 8,000 markers that differentiated these 450 samples that we sequenced using this panel. And typically, if you look at pennycress literature, you see only two major groups that forms. One is a vertical line that you see on the right, which is usually North American and European cohort, and extreme to the left is usually an Armenian cohort. And over the years of breeding and how we've introgressed some of the material that has been collected internally, what we are really seeing is we are creating subgroups or groups that are diverse from the North American or European panel or the Armenian panel. And this is very encouraging. And using this panel, we can highlight the fact that we are moving away from wild germplasm and creating varieties that are going to be unique to CoverCress product portfolio. We continue to work on this data. We are using this data to understand the linkage disequilibrium, really defining those new populations that are getting formed as the breeding continues. And then we'll use this to leverage for future genomic breeding efforts. Finally, when you start thinking about having the right genotyping platform, you start thinking about, can you use that platform for performing genome-wide association analysis? So we wanted to see if this data could help us dissect a trait that we've been collecting in our program over the last years. As you know, we've been working on gene editing as one of our methods to deploy the traits. We've been transforming a lot of lines over these years, and we see there is a lot of variation and genotype dependency for transformation rate when subject to floral dip method. And this is not unique to pennycress. Most of the species have genotype bias towards transformation rate. We wanted to see if these variations can actually be mapped, because if the trait can be mapped and a marker can be identified, it could eventually be fixed in a lot of our germplasm to increase our ability to deploy multiple traits in a very short timeline. So we took this phenotypic data and used the genotypic data on the lines that have been subject to transformation. And we were able to use that data to really map QTLs in a few regions of the genomes. And this was very exciting. Obviously, it's a preliminary data. There needs to be more work done on this to validate these QTLs, but finding genes or markers that could impact transformation rate could be huge and may be applicable for other crops in the future as well. So with that, I would like to summarize by saying that identifying this genotyping platform, especially with the FlexSeq that covers 5,000 unique regions in the genome, has helped us perform genetic mapping. Hopefully, we can use this for genomic selection in the future by fingerprinting lines at different generations or training population, improve our quality of germplasm by being able to track the markers generation after generation as the products move into the portfolio. With a platform like that, we anticipate more data accuracy, reproducibility, and the consistent coverage of the genome allows us to see where the linkage blocks are being formed or how the selection biases are occurring in the breeding program. There's a lot of application, genomic selection, QC, and really excited that having these genotyping platforms would be key for the future CoverCress product. With that, I would be happy to take any questions you may have. Thank you for listening to my talk. Appreciate your presence during this session.
Thank you, Dr. Chopra, for your informative presentation. Before we jump into the live Q&A portion of the webinar, there is a brief poll question. If you could please participate in that. It's located in the top left of your screen. Take a moment to just submit your response there. It's either yes or no to would you like to request a quote, a demo, or speak to an expert. Select your answer, scroll down, and click submit once you have had a moment to do so. Let's go ahead and get started with the live Q&A portion of the webinar. All right. Our first, if you have any questions you'd like to ask Dr. Chopra, please do so now. Take a moment to type your question into the Ask a Question box and click Submit. We'll answer as many questions as we have time for. So our first question that we have here is, given the demand is so big for renewable diesel, what other alternative crops are being discussed, or do you think CoverCress will be able to meet these demands?
Oh, thanks, Cassie. Like I mentioned earlier in my talk, there's a lot of efforts throughout the country thinking about different feedstocks, and there's camelina, carinata, venture canola. All of these are going to be critical to meet the demands that are growing due to the shift towards low carbon fuel. CoverCress will be a major contributor, but we will need more than one crop on the landscape to address the challenge that we are seeing today.
Great, thank you so much. Our next question that we have here says, there is a good translation from Arabidopsis for many of the traits you discussed here, then wouldn't editing be a best pathway for everything, every change you needed?
That would be really great if it could be that easy. You know, it's a different species. There's a lot of correlation. We can convert traits from Arabidopsis to Phenicrus for the most part, but there are some uniqueness, and that's why we need to do discovery work. We need to use the platforms like we are developing here and identify genes and areas that would make CoverCress as a better crop. And that's why editing won't be the only solution for the crop improvement.
Great, thank you. And again, if you have any questions that you'd like to ask our speaker during the live Q&A portion of this webinar, you can do so by typing them into the Ask a Question on the lower left portion of this screen and clicking Submit. We have another question here. It says, what are the benefits of having a genotyping platform for CoverCress beyond breeding applications?
So genotyping platform is useful for many reasons. I talked about like how it can be applicable for different purposes. But one of the things that we find it very valuable as we are able to edit multiple products from multiple genes and the germplasm that's kind of moving through the pipeline, and the seed is so small. There's always a challenge of keeping the purity, and as we are continuing to build this product portfolio, a platform like this would help us use the QC data and say, here is the line that you expected it to be, and that's what is getting edited rather than a random seed that got popped up from prep methods. So I think a system like this is going to be useful hugely even in the quality control aspect of deploying our products into the market.
Great.
Thank you.
It looks like that is all of the questions that we have in our queue so far. So I wanted to thank you again, Dr. Chopra, and ask, did you have any final comments for our audience here today?
Yeah, no, thank you everyone for attending the session today. I appreciate LGC for letting us present the CoverCress story, or the domesticated pennycress story that's been developing over the decade. And I'm happy that we are continuing to see this progress towards what would be necessary for the second generation products for CoverCress or domesticated pennycress. And relationship like this are going to be crucial, as we can also have relationship with academic partnerships like groups like I Prefer, I Prep. All of these are great partnerships to bring a new crop to the market more quickly than doing it by yourself for a small company. So feel free to reach out to me with any questions you may have. You can find my details on the website, or thank you. Thank you for your time.
Thank you again, Dr. Chopra, for your time today and for your important research. We would also like to thank LabRoots and our sponsor, LGC Biosearch Technologies, for underwriting today's educational webcast. Before we go, I'd like to thank the audience for joining us today and for their interesting questions. Questions that we didn't have time for today and those submitted during the on-demand period will be addressed by our speaker via the contact information you provided at the time of registration. This webcast can be viewed on demand. LabRoots will alert you via email when it's available for replay. We encourage you to share that email with your colleagues who may have missed today's live event. Until next time, take care, everyone. Goodbye.